So this part of the class is devoted to kernels that have been designed for biological sequences. So you will see that there is a very rich literature about such kernels to deal with sequences. And what we are going to do in this lecture is to only go through a subset of these kernels. We are going to do, let's say, a very short introduction. But if you are interested in the topic, you will see that we have many slides and many kernels that have different interpretations that model different phenomena. So it's a very interesting literature, which I will encourage you to read if you are interested in that. So first, let us do a very brief introduction about the motivations for genomics, for machine learning, for sequences. And in terms of history, I'm not the best person to do that, but there are a few things that you should know that probably you already know. The history of genomics is pretty old, and there are a few milestones that are pretty famous. For example, 1953, the structure of DNA has been discovered by Crick, Watson, Wilkins, and Franklin. So we have here 3 men, 1 woman, and the 3 men got a Nobel Prize. Don't ask me why. Then later, something that made lots of noise was in 2003, the Human Genome Project was completed. And this opened the way to a huge amount of work to process genomic data. So just if you don't remember your classes from high school, this is a cell, various components of the cells. And for many of them, we don't care much about them, but there are a few elements that we care about. We'll care about the nucleus, what is inside the nucleus. We'll care about the ribosomes that are used to translate RNA into proteins. What are chromosomes? Chromosomes are simply, for us, they will simply correspond to the DNA code that is present in the cells, in the nucleus of the cells. And of course, you know that for humans, there are 23 pairs of chromosomes. Okay. What else? What is, so here is simply a picture that show you the structure, the famous double helix structure of the DNA, which was also appears in this famous picture by Watson and Crick, who received, like I said, the Nobel Prize for this discovery in 1962. Even so, real key to this discovery were the X-ray crystallography images obtained by Rosalind Franklin, who was simply forgotten in some way by the Nobel Prize Committee. And here to give credit to Rosalind Franklin, here is a picture of her. Okay, so the double helix, well, it's for us, what we will simply see, we'll see DNA as a sequence of characters, DNA bases. We have 4 different characters, G, C, A, T. And this double helix will code the DNA into 2 ways, because there will be a correspondence between the base G on the left and C on the right. You see C and G here. And similarly, A will be associated to T on the right. Okay. These base elements will simply be related by this hydrogen bonds. So I don't know much about this, so I don't want to comment much of this. I will simply take my computer scientist hat, and I will be very happy to see that there is a code that represents genomic information. So something that you should already know is that what DNA is for, it's for coding proteins. And since the elements that code proteins do not have access to the nucleus of the cell, there is actually small fragments called RNA or messenger RNA that will copy parts of the DNA, move out of the nucleus, and then will be read by ribosomes that will produce proteins. So it will be a bit an oversimplification to say that RNA is just a copy of DNA. In fact, it's a copy of fragments of DNA in a specific order. So that's not really the topic of this class, but this is basically the phenomenon of interest, how to code proteins that are the basic elements of life. And if you don't know all of this, I really encourage you to just read the newspapers, because you can see these sort of things every day, given the context today. Okay, proteins, what they are sequences of amino acids. And I will come back later to amino acids, but for us, we will see amino acids as characters in some alphabet, which is larger than the alphabet of the 4 DNA bases. So it turns out that in this particular part, we are going to see proteins as sequences, but in fact, what turns out to be very important with proteins, and again, you can see that in the newspapers, is that the protein sequence, amino acids, have particular 3D representations in the real world, such that a natural representation of proteins seems to be more a graph than a sequence. Nevertheless, processing the sequence data is still very important. Okay, so what are alphabets I was mentioning? So we have the 4 DNA letters, A, T, C, G, that are transcripted into RNA. We have 4 letters. For some reason, we have a U here instead of T. These are simply different elements, but there is a one-to-one correspondence between them. Like I said, RNA is just not a basic copy of DNA. It will simply take some particular fragments of DNA and assemble them in some particular order. Such that, in fact, if you take a gene in DNA, it can give rise to several types of proteins. Anyway, at the end of this process, we have proteins that are coded by on an alphabet of 20 letters, which are the 20 types of amino acids. So that means that to code an amino acid, you need at least 3 DNA bases, which we call nucleotides. And we have here simply a table that explains what is the correspondence between the type of amino acid, these small elements in the table, and the bases, the nucleotides, basically. Okay, I will go fast on this because this is a machine learning class. This is not a class about biology, and I'm not very legitimate in terms of teaching biology anyway. Now comes the genetic sequencing technology, which produces lots of data. So the Human Genome Project, which started in the 90s, the goal was simply to sequence the 3 billion bases of the human genome. So if you take our pairs of chromosomes, there are about 3 billion of bases. It's actually not that much if you think about it. Anyway, finding what are the bases in the human genome, it doesn't mean that we all have the same exact genome. There are small variations. It means what we have in common among the humans. Anyway, this was a big deal to sequence this genome. It was a consortium with 20 labs, 6 countries, and the cost was between $0.5 and $1 billion. So that was very expensive. And after more than 10 years of work, this was the big deal. The human genome the sequencing was completed, and there were lots of interesting findings. For example, we realized that there were only 25,000 genes, which is only 1.2% of the genome, which means that what the rest of the genome was doing was just unknown at that time. It doesn't mean that everything is solved yet, but it means that a large part of the genome at this time was considered to be junk DNA, meaning DNA which is useless. It turns out that there are new discoveries that explain that part of it is not junk. There are also phenomena that are for which this DNA is useful. Anyway, here we are going to focus on the genes, and one of the goals will be to be able to do machine learning with this genomic data. Okay, one slide about the cost of genome sequencing. This slide is actually not updated. I forgot to update it. It was from 5 years ago. But basically, the cost of sequencing a genome has decreased tremendously. Here, it was costing $100 million in 2001. In 2015, it was about $1,000. It turns out that on more recent plots, this cost has been relatively constant here. Something I should also say is that this plot is not completely fair because the quality of the sequences that we get is not exactly the same. These very expensive technologies were producing also very accurate genomic data, which is not the case for the cheap ones. But what is still really striking is that we went from $100 million to $1,000, meaning that now we can produce lots of genomic data and we need tools to process this data. Protein sequences, yes, this is just the alphabet, right? That shows the names of the different amino acids. And what are the challenges? Well, a protein sequence can be seen as a sequence over this alphabet of 20 letters, and the size of the sequence can vary from a protein to another. This one, for example, is the sequence of insulin. And we need algorithms to analyze, classify these sequences. So I'm not going to give you many examples of applications, but you should easily find this in the literature. Well, just one basic example. We may want to know, given sequences of amino acids, if the protein is going to be secreted in a cell or not. And to build a classifier that is able to tell whether it is secreted. So it will be a label +1 or not, it will be a label by -1. Meaning here we simply justify a basic example. We simply justify the need to do binary classification here. Now let us go to the more technical part. So given what we have seen in this class, what can we do to do machine learning with sequences? There is a first approach, which does not require kernels, which simply say, well, each sequence, I want to represent it as a vector, phi of x, so the sequence is x, that lives in some space f, and I want simply to train a classifier for vectors. So basically, let's say a linear model. And for that, you can do many things. After you embed your sequences into a vector, you can do nearest neighbor, encode a linear perceptron, logistic regression, support vector machine, ridge regression. Now, the question is, what is a good embedding for these sequences, and how do I do all of this efficiently? So it turns out that the kernel methods for protein sequences have been widely investigated since the end of the 90s. So this corresponds more or less to the time where genomic data started to be available. Even so, the full sequence of the human genome was available only in 2003. And if we want to use kernel methods, there are questions that are, well, what is how can I design a kernel which is positive definite for sequences? I want this kernel to be fast to compute because if you give me a kernel that takes forever to compute, it will be hard to use, which is also adapted to the problem, meaning that what is a good kernel? A kernel encodes a similarity function between sequences, and a good similarity has to be defined given domain knowledge. Meaning there are physical phenomena, biological phenomena that we need to model. Then comes this part where we are going to do kernel engineering for protein sequences. Here I will very briefly review some of the kernels, give you a few words about each of them, but we are going to focus in this class on only a few of them. And there are also many slides that I'm not going to comment that will also talk about this other kernel. So feel free to look at them, if you're interested in that. Okay, so sorry, the first class of kernels correspond of defining vectors that represent our sequences, but vectors that are very high dimensionals, meaning that it will be simply hard to manipulate these vectors explicitly, such that we will need to use a kernel method. Basically, we are going to represent sequences as very high dimensional vectors, and to compute the inner product, meaning the kernel function, we'll need to design clever algorithms. So we are going to talk in particular about the spectrum kernel, the mismatch kernel, and briefly talk about the substring kernel. You'll see what this means. It turns out that something I have not presented in this class, there are ways to design kernels based on a probabilistic model, which is a generative model of the data. Meaning, if you have a probabilistic model, a likelihood that represents your data, there are natural tools to do that. Again, this is a part which I have not presented here. Maybe I will produce a video about them later. But at this point, if you're interested, you should look at this section from the slides that are devoted, for example, to the Fisher kernel, the mutual information kernel, or the marginalized kernel. The Fisher kernel is particularly interesting for me because, in fact, when you look at the computer vision literature, so these are not sequences, but in computer vision to represent images, we have also lots of probabilistic models. It was giving representations of images that yield state-of-the-art results for classification just before 2012. If you know a bit about the history of computer vision, in 2012, convolutional neural networks started really to take the lead on image representation on almost all tasks that you may consider in computer vision. But just before that, Fisher kernels were really the way to go in computer vision. So that's why they are interesting, at least from a historical point of view. But in fact, there are also many other contexts for which you cannot train a convolutional neural networks, for which you have a generative model, and for which Fisher kernels are interested to consider. There are also kernels derived from existing similarity measures for biological sequences, which are not positive definite. And we need to work a bit to make them a valid positive definite kernel. So this work yields the local alignment kernel, but I'm not going to talk about this in this video. Okay, so kernels derived from large feature spaces. So the idea is to represent each sequence X by a fixed length numerical vector. So this is important that the representation has the same length such that we can train a basic linear model on it. So phi of X, the representation of the sequence, is now a vector in Rn, and n may be very large. Let us skip this and jump to this popular approach that consists of enumerating small patterns that are related to the sequence of interest. So consider our alphabet A. If so, this notation here, a to the power of k, represents the patterns, meaning the sequence of letters of size k, meaning simply a pattern of k different letters. And phi of x will be a very high dimensional vector whose size is exponential in the size of the alphabet, that contains a value for each possible pattern u of size k. So x is a long sequence, u is a small pattern of size k, and phi u of x is simply a quantity that tells us if this pattern u is related to x in some way. And I will be more precise soon about what I mean by related. So let us be more precise indeed. What can be phi u of x? The simplest thing we can imagine is that phi u of x can be the number of occurrences of u in the sequence x. So basically at the end, it simply means that if in your sequence you have u that appears 3 times, phi u of x is equal to 3. And it yields a kernel which is called a spectrum kernel. For those of you who are also familiar with computer vision or familiar with the processing text data, classical approaches before people were using deep learning was also to use such representations where we will count the number of occurrences of words in a text or of local features in images. So here this is relatively similar. At the end, what is phi of x is we consider the spectrum kernel. It is a sort of histogram where each entry in the histogram counts the number of occurrences of a particular subpattern. So of course, this is a very high dimensional vector. Now, it turns out that for biological sequences, at least for DNA sequences, this representation here lacks some flexibility. Because imagine that if you have a large pattern U, which is relatively large, let's say it has 10 letters, you will have a value phi u of x that is non-zero only if this pattern appears exactly in the sequence. But it turns out that for many types of data, it's actually very useful to relax this definition and to count that a pattern U will appear in X if we can see U in X up to some mismatch. For example, if this M here is equal to 1, we'll say that phi U of X, that U appears in X if we can see the pattern U in X by allowing to change one letter, up to one letter. And this gives a kernel which is very effective, which is called a mismatch kernel. For many problems, this gives better results than the spectrum kernel, which is not flexible enough. Then there is also this idea that, well, perhaps modeling mismatch is not enough. Perhaps we should also consider that U can appear in X, even if there is a gap in the pattern U. You will see with examples more precise statements about what this means, but the motivation was coming from biological phenomena where in biological sequences we can have DNA letters that are inserted at some positions, or sometimes DNA letters that are deleted. So that would motivate the need to allow gaps when in the definition of occurrences. You will see what I mean by gaps very soon. So before that, let us consider the spectrum kernel and let us see a few examples such that we get a bit more intuition about what this actually does. So if we consider this sequence X, which is a sequence of amino acids encoded by their respective letters, if we consider all of the patterns of size 3 that appears in this sequence, we can simply enumerate them. They are at most, the number of such patterns is bounded by the length of the sequence. We have, for example, CGG, which appears here, GGS, and so on until FGV here. And then what is the spectrum kernel? More precisely, the K spectrum kernel is simply the inner product between the vector phi of x and phi of x prime for two sequences x and x prime. This can be written as the sum over all possible patterns U of phi u of x times phi u of x prime. So you see that to have an element which is non-zero in the sum, we need to have a pattern U which is common both in x and in x prime. This perhaps gives a bit more motivation about why we need to introduce the concept of mismatch kernel later to relax this definition, because here sometimes it's easy to build when k is large enough, it's easy to build sequences that have lots of things in common, but for which the number of patterns of length k in common is 0. But if you allow mismatches, this can change drastically. Okay, so let us talk a bit about computation. So this computation is formally a sum over an exponential number of terms. But we know that in phi of x, we have at most, so this represents the length of the sequence. So it will be the length of the sequence minus k plus 1, so more or less the length of the sequence. Terms, entries in phi of x that are non-zero. It turns out that if k is not too large, for example, if the total number of patterns can be coded in 64 bits, for example, which is reasonable for various, basically it means if this quantity here is smaller than 2 to the power of 63, it means that it's easy to implement on your computer a method that will do the computation in a complexity which is linear in the length of the sequences. This is something which I think is very important for you, especially if you are dealing with a data challenge that requires to process biological sequences. So this is typically a point which we can discuss in details in the interactive session. But assuming that your phi of x anyway, you know how to compute your phi of x for every u. You have enumerated all of the possible patterns and you want to do classification. If you have a linear model on phi of x, which is represented by this vector w, then this is simply a sum of at most this number of terms, meaning the complexity to compute the prediction is simply also linear in the length of the sequence, which is a nice property. So this principle works with any types of strings. It's relatively scalable. And like I said, there are important variants that allow to code mismatches. So if you cannot do the pre-indexation of these strings, meaning if this quantity here requires the number here requires too many bits, then there exist particular structures, data structures that allow to have also relatively fast classification. So I'm going to mention this first one, which is called a retrieval tree. In the literature, it's also called a trie. That allows to compute the kernel entries k of xx prime in a complexity which is linear in the length of sequences times k. So this is larger than this by a factor k. I must also say that, in fact, in practice, it's much larger than if you can do the pre-indexation, simply because the big O here is hiding a small constant here, and here it may hide a larger constant. That means that basically I will never encourage you to code the spectrum kernel using a retrieval tree. I would really encourage you to code it using the pre-indexation of the strings, hoping that the number of possible patterns that you consider is not too large and that you can do this pre-indexation. Anyway, I find, nevertheless, this data structure to be interesting. So let us look at what it's doing. So what the retrieval tree is doing is that it's a tree that has a root and that has a depth which corresponds to the size of the patterns that you consider. And the leaves of the tree correspond to all of the patterns that appear in the sequence. For example, if I take GTA, turns out that GTA appears here at the end in my input sequence. So we are going to code each sequence as a retrieval tree that in fact enumerate the possible subpatterns. So how do we proceed? It's actually relatively easy. We start by a first stage here that has one node for every letter that appears in the sequence, ACGT here. Then at the next level, we enumerate here the patterns of length 2 that appear in the sequence. So they necessarily start with A. So when we have A, we have then AC appears, AA appears, and so on. And we proceed until the leaves that such that if you start, for example, from GT, you see that in the sequence we have GTT and GTA only. So GT gives 2 nodes, GTA and GTT. It turns out that it's relatively easy to see that you can produce this retrieval tree, to build it in a complexity which is equal to the number of nodes, meaning k times which is at most k times the length of the sequence. It's relatively easy to, if you are not convinced by that, you can simply, take some time at home to be convinced that indeed you can do that with this complexity by simply scanning the sequence and producing the nodes each time you encounter a new pattern. So that's not too complicated. Anyway, once you have coded a sequence X like this, if you have then coded a sequence X prime in a similar fashion, we have 2 different trees and that have some nodes in common. And in fact, how do I compute this quantity k of xx prime? I compute it by enumerating the patterns that are common to the two sequences. And we can do that by doing a joint exploration of the 2 trees, meaning we are going to follow paths that appear in both sequences. For example, assume that AAC is present in X and present in X prime. It means that both of them has the same edge that goes to A. Both of them have the same edge that goes to AA. And both of them have also this edge that goes to AAC. So you see that you can do this joint, this simultaneous exploration of the trees. You enumerate all of the possible paths, and at the end, you will simply keep the leaves that are common with the two trees. So that gives you this complexity. But again, an important message is that you can do something more clever by associating a unique number in 64 bits to each pattern. And then playing a bit with the data structure, which is nothing else than a sparse representation of a vector. For example, you can use SciPy for that. At the end, you will get a better complexity to compute your inner products. And again, let us keep that for the interactive session. Something I will skip is that it turns out that there are also more clever data structures that are a bit more complicated to present. It's called the suffix tree that will give a better complexity, which is the same as pre-indexation, but with a much larger constant value, which is hidden in this big O notation here. But I don't want to spend time on that. What I want to do is to discuss this other kernel, which is called the substring kernel, the one that goes beyond encoding mismatch. Because, okay, by the way, everything I said before about enumerating patterns, you can do the same with mismatch, meaning that you will add leaves to your retrieval tree by allowing you will add when you do the pre-indexation of sequences, you can add also the sub patterns that appear up to one mismatch, and it will not change much in terms of methodology. Now, what will change? What will lead to more significant modification is the concept of gaps. So what is, and to do that, we introduce a few notation. So the notation we'll see they will become intuitive very soon in the next slide when I give an example. But instead of considering simply the position of a pattern in the sequence, now that I will allow gap, I will need to consider positions of every letter of a pattern. If I have a pattern of size k, I will consider also a sequence of indices I1 to ik. And for a string x, which is x1 to xn, if I consider a sequence of indices of size k indices, I will now define a substring as simply the concatenation of letters that appear at these different indices. Okay. And what is the length of the substring? The length will be simply, will not be only k, the number of characters, it will also depend on the indices. Okay, it will simply be ik minus I1 plus 1. Let us give an example to fix all of this, because I admit it can be a bit confusing at this point. If you consider the sequence abracadabra, if I consider this indices 3, 4, 7, 8, 10, then the substring is radar. Indeed, 3, 4 are A, 7, 8 DA, 10 R. And what is the length of this substring? It's not 5, this is 8. It is, when I look here, I have 1, 2, 3, 4, 5, 6, 7, 8 letters. So this is a substring of size 8 with 3 gaps, C, A, and B, and this is what I mean by gaps. And now this gives this new kernel that will assume that I will consider that radar is part of abracadabra with 3 gaps. Now, of course, it will not be fair. For example, let us come back. It would not be fair to say that radar appears once in the sequence and that dabra appears also 1. Why? Because dabra appears exactly 1 here without any gap, and radar requires 3 gaps. So basically, it means intuitively I want to put a larger weight on dabra, and I want to put a small weight on radar. This can be encoded using an exponentially decreasing factor that will simply decrease with the number of gaps. So now, what if I consider a sequence of k letters u? What will be phi u of x? Phi u of x will simply sum all of the possible set of indices, of k indices, and it will check if x, the substring x of I, is exactly equal to the pattern u. And there will be a term here, which is if this is the case, we will also have a term that decreases exponentially fast with the length of the sequence, which depends on the number of gaps, of course. And at the end, our substring kernel is simply the inner product between phi of x and phi of x prime as before. So you see that compared to the spectrum kernel, it seems that we have made our life very complicated because we have this sum which is large. And here we have this sum which is also large just to compute a single entry of phi of x. And in fact, because there is this relaxation of that allows to have gaps, instead of having a very sparse vector phi of x, the number of non-zero entries in phi of x will be much smaller than with the spectrum kernel. Here is a basic example of what is phi u of cat for different values of, phi u of different sequences, cat, car, bat, and bar, for different patterns u, CA, CT, AT, BA, and so on. Okay, so question, does CA appears in cat? Yes, it appears with 0 gap. So it means that given the previous definition, so by the way, lambda is a non-zero parameter which is between 0 and 1. So CA appears in cat and the length is 2, so I get lambda squared. CT appears also in cat, but with 1 gap, so the length is of this substring is 3. So I will have here lambda to the power of 3. AT and so on is lambda square, and then BA does not appear at all, so I get zeros here. So I can do that for every row. And now what I can do when I will compute the kernel between 2 sequences, what I will simply do is simply compute the inner product between a row. So here, the row cat is here. So basically here, this row is simply phi of x, where x is equal to cat. So I compute the inner product between phi of cat and phi of cat. That gives me what? That gives me lambda 4 plus lambda 6 plus lambda 4. Now, if I want to compute the kernel values between cat and car, so cat is here, car is here, it turns out that in common, they only have CA. So I will have lambda squared times lambda squared. That gives me lambda to the power of 4. And what about cat and bar? Cat, bar, they don't have a single pattern in common. So the kernel value is equal to 0. So far, it looks like it's quite a desperate way of computing these embeddings, because, well, the size of phi of x can be very large, because here this is a basic example with sequences of length 3 with patterns of size 2. Of course, we are interested in proteins. In DNA sequences, we may have sequence of size 500 and patterns of size 10. Of course, we cannot do something like that. Something which is nice. So this is here exactly what I said, which is if you plug the previous formulas, you would get an enumeration which is just intractable. Now, it turns out that there is something nice going on here, which is that this previous computation has a particular structure. And this particular structure allows us to compute the kernel value as a function of other kernel values involving smaller sequences. So there would be, in fact, a recursive formula that we can derive that allows you to compute the kernel value k of x, x prime for 2 sequences using dynamic programming in a polynomial time complexity. So this is here that would be horrible to present precisely in these slides, but the formula are, in this video, but the formulas are on the slides. If this is here the definition of phi u of x, we sum over all the possible set of indices. We have this each time x of I is equal to u, we obtain this contribution lambda to the power of the length of the index set I. We can introduce other quantities, I'm not really telling you why, that are slightly different. Here you simply see that instead of ik, here I get here the length of x. We can define, observe that there are recursive relations. If you have, for example, if you know how to compute psi v of subsequences of x, so this represents here the first part of x up to letter j. If we add a letter, then we have a sum which is reasonable in terms of computation to obtain. So there are recursive formulas appearing here. There are also other recursive formulas that you can obtain. So here I must precise that I'm not asking you here to follow all of the different steps in your head here. If you want, you can spend more time at home to do it on a sheet of paper. But given these previous definitions, phi u of and psi u, it turns out that if you add a letter to A, if the last letter of u is not A, then we have this relation. We also have this other relation that relates psi u of x plus a letter to psi u of x without this letter. If the last letter of u is A, we also have a relation that relates phi VA of XA, and these quantities that each time involve a smaller quantity. You see that here, instead of VA, we have V. Instead of XA, we have X. So this is a shorter sequence. So you see that the goal of dynamic programming is to efficiently build the solution of a large problem given the solution of a slightly smaller problem, which is itself built from the solution of a slightly smaller problem, and so on. And at the end, compute all of this with a reasonable complexity. So we have this sort of relation, and now we can also consider again our kernel here, which involves the quantity phi u of x. We can also consider this alternate kernel bk that involves these alternate quantities psi u of x. Remember that psi u is a tiny modification of phi u, but which is related in some way. And if you work a bit more, you can obtain a recursive computation of this kernel bk of x plus a letter and x prime as something that depends on bk of x prime without this letter, plus something that depends on bk minus 1 and also smaller sequences, in a complexity here which is of size at most the length of the sequence. If you continue that, this is the final formula which is of interest. It turns out that this quantity bk, if you add a letter to x and a letter to x prime, then you see that you can obtain it in constant time given quantities that you may have computed previously, which are bk of x prime b, bk of xa x prime, bk of x prime, bk minus 1 of x prime. This quantity bk, we'll call that the dynamic programming table. This is something that we are going to fill. Okay, now here is the idea that you may want to get if you want one day to implement this. You start with the first table b0. So what is bk, by the way, for two sequences of size n and n prime? bk minus 1 until b0, they are tables of size n times n prime. And what you want is for each entry, you want to compute, for example, if I consider the entry m and n prime of the table bk, I want to compute bk of the first m characters of x and the first m prime characters of x prime. And I can fill this table recursively with each time with a constant number of operations. If I do that in the right order. So this is nice because it means that the complexity to fill this dynamic programming table is simply the number of entries in this table. So the number of entries in bk is the size of x times the size of x prime. And I have k of such tables. So the total complexity is this. Now, if I come back to the previous slide, so that was not here, that's the next one. Now, what is nice is that these quantities, this dynamic programming table bk, it turns out to be useful to efficiently compute what is of interest for us, which is the kernel value k. So you see that these are steps that are pretty boring, but I guess that you get the intuition. If you have a lot of motivation, you can try to rederive that by yourself. I must say that this is something I did once in my life when I was updating these slides, doing that in the train. So I hope that these formulas are correct, but at least they give the right complexity. So in summary, the complexity for these kernels for spectrum and mismatched kernel is simply linear in the size of the sequences. So this is fast to compute. This is an implementation which requires to do some pre-indexation of the different patterns that appear in the sequences. If you cannot do that, you have this complexity where you pay a factor k. You can also remove this factor k if you use a suffix 3, but this is an implementation which is a bit more complicated, where you pay a price in the constant here in the big O notation. If you want to do substring kernel with dynamic programming, the complexity is larger, but this is the price to pay to model gaps. So the complexity is now the product of the size of the sequences times k. And at the end, the feature space has a very high dimension. But even so, the dimension of phi of x is exponential. We have these algorithms that compute the kernel values in polynomial time and even very efficiently for the spectrum and mismatch kernel. So remember, something important is that we are sending data to a very high dimensional space. It also means that we need to regularize if you want to prevent overfitting. So that's it for the, I don't want to, so this is already hard to digest, these three types of kernels. But for me, what I find particularly interesting here is that we are kind of forgetting a bit the mathematical aspect here. Here we really need concept from computer science. So this is what makes these kernel methods pretty rich, is that we have concept from mathematics. If you look at the section on Green, Bochner, Fourier kernels, and here we need concept from computer science. Something you will also see if we look at the lecture on graph kernels. Kernels represent graphs. They are related to graph theory. This is also a very interesting part. Now, what is left, what I'm not going to comment now, is many other types of kernels for sequences. And but really just feel free to spend time at home looking at them if you are interested in that. In particular, there are the kernels derived from generative models that I have mentioned already. Other interesting kernels derived from a similarity measure, and we discuss a few applications here, but in fact, probably we could also update it with more recent applications. Okay, thank you for your attention, if you managed to follow the video until this last minute. And next time we are going to discuss, in fact, we are going to discuss this other types of kernels that involves more mathematical aspects.